Hello everyone, so today I'm going to present on one of the earliest antibiotic discoveries called Bacitracin, which usually used for topical infections. So let's go back to where it all began, where the discovery of Bacitracin was first isolated in 1945 during a time when scientists were still exploring soil bacteria as a potential sources of new medicines. Interestingly, it was discovered from a wound infection on a young girl named Margaret Tracy. A strain of Bacillus subtilis was found in the infected wound, and researchers noticed it was producing something that killed harmful bacteria growing around it. They extracted this compound and named it Bacitracin in honor of both the bacterium Bacillus and the girl Tracy. This discovery was of many examples of how nature, especially soil microbes, can be a rich source of antibiotics. After its discovery, further research revealed that Bacitracin is not only produced by Bacillus subtilis, but also by Bacillus licheniformis. This bacteria can be grown in lab conditions through fermentation, which allows for a large-scale productions. Over time, Bacitracin became widely used, especially in topical treatments for skin infections, due to its strong activity against gram-positive bacteria. Now, let's look at the structure and properties of Bacitracin. Chemically, Bacitracin is a cyclic polypeptide antibiotic. This structure includes a thiazole ring, which helps it bind specifically to its bacterial target, making it effective at blocking cell wall formation. Physically, Bacitracin appears as a white to pale buff powder. It's hygroscopic, meaning that it absorbs moisture easily from the air. In terms of solubility, Bacitracin dissolves well in water and ethanol, making it suitable for creams and lab use. It's very stable under proper storage conditions, but it's sensitive to moisture and heat. Moving on, let's talk about how Bacitracin mode of action works to kill bacteria. So, it involves inhibition of cell wall synthesis. Bacitracin targets something very important in a bacteria cells, which is the cell wall. The cell wall gives the bacteria its shapes and protects it from bursting. So, to build this wall, bacteria use special building blocks called peptidoglycan precurses. These are made inside the cell and need to be transported to the outside, where the cell wall is located. But these precursors can't move on their own. They need help from a carrier molecule called bactoprenol. Its function is to pick up the peptidoglycan pieces from inside the cell, carries them across the cell membrane, and delivers them to the outer side so the wall can grow. Once the delivery is made, the bactoprenol needs to go back inside the cell to pick up more building blocks. In order to do that, it must undergo dephosphorylation so it can be used again. So, this is where Bacitracin steps in, where Bacitracin binds to the bactoprenol and blocks this dephosphorylation step. As a result, bactoprenol can be reused. That means that the building blocks stop being delivered and the construction of the cell wall comes to a halt. Without a proper wall, the bacterial cell becomes weak and eventually bursts and dies. This action is especially effective against gram-positive bacteria like Staphylococcus aureus and Clostridium difficile because they rely heavily on their thick peptidoglycan layers and don't have the extra protective outer membrane that gram-negative bacteria do. For resistance against Bacitracin, although Bacitracin is very effective, especially against gram-positive bacteria, not all bacteria are easy to kill with it. In fact, many gram-negative bacteria are naturally resistant. This is because they have an extra outer membrane that acts like a strong protective shield. It stops Bacitracin from getting inside the cell and reaching its target. Bacteria like Klebsiella pneumonia and Pseudomonas aeruginosa fall into this category. Even if the Bacitracin is present, it can't get through that outer layer to do any damage. But resistance isn't just about blocking Bacitracin at the surface. Some bacteria have developed ways to survive even if the drug gets inside. One method involves a flux pump like the BCR-ABC systems that act like a tiny pump that pushes Bacitracin back out of the cell before it can do any damage. Others use enzymes to bypass drug effect. A good example is BACA protein, which helps reset the bacterioprenol carrier even when the Bacitracin tries to block it. Even more concerning is the spread of resistant genes, such as the BCR-ABDR cluster. These genes can be shared between bacteria, especially in an environment where antibiotics are used heavily, like animal farms. Now, let's talk about the cause and how Bacitracin is used for today. For everyday use, the Bacitracin is most commonly found in the topical ointments, especially in combination with other antibiotics like neomycin or polymyxin B. These ointments are available in pharmacies and usually cost between 40 ringgit to 65 ringgit, depending on the brand and size. They are used for minor cuts, burn, or skin infections and are considered safe and effective when applied externally. In research settings, Bacitracin is also valuable. Laboratories use research-grade Bacitracin powder for microbiology testing or antimicrobial assays. There is also an injectable form of Bacitracin, but this is rarely used. It's typically reserved for serious hospital infections when other antibiotics are not effective. So, in the QR code, there are a list of references that I use. Thank you so much.